Hi guys, this is Kumar. Today we are going to learn how the cells divide in our body, and that process is called as cellular division. So in the cell theory, you have studied that all cells are derived from pre-existing cells. So our life, which started as a single cell, as a zygote, and as an adult, we have 10 trillion cells. So how these cells are coming from a single cell to 10 trillion cells, and that's through a cell division. New cells are produced from the growth and to replace the damaged or the old cells. So if there are 10 trillions of the cell, they are having replaced by new cells, and the cells which are damaged, they are also being replaced. So the process of the cell division is different in prokaryotic, which are bacteria, and the eukaryotic, that is protists, fungi, plants, and animals. And now you already know the difference between the prokaryotes and the eukaryotes, which is a major difference is the nucleus. So how to keep the cell identical? Because in our body, most of the cells are having the same identity. So the instruction for making a cell parts are encoded in DNA. So how the cells which are coming from the pre-existing cells are having the same information, and that information is encoded in DNA. So such new cells must get a complete set of the DNA molecules from the pre-existing cells. So how do they get these DNA from the pre-existing cell is through the cell division. So the DNA, which is having the information inside the cell, is an important part that it should be handed over to the next dividing cells, which has all those information how the cell metabolism is going to take place. So the DNA must be copied or replicated before the cell division, so that when after the replication it is passed down to the next dividing cell. So each new cells will then have an identical copy of the DNA. So in this picture you can see this is a parent cell. After dividing, it got into two identical daughter cells. So daughter cells are those which are the cells which came from one parent cell. So if one parent cell is divided into two, the two cells are called as the daughter cells, and they are identical. How? Because they are having the DNA which was passed down from the parent cell to the daughter cell. Chromosomes, which are the important part of the cell division. So what's the basic difference between a DNA and chromosomes? Let's take a different. So let's start with the prokaryotic cells. Prokaryotics do not have a nucleus, and the DNA of the prokaryotic like the bacteria is a one circular chromosomes which is attached to the inside of cell membrane. Because there is no nucleus, the DNA is exposed out without any boundaries. And talking about the eukaryotic chromosomes, all the eukaryotic cells store a genetic information in chromosomes. And most of the eukaryote have between 10 and 50 chromosomes in the body cells. But talking about the humans, as a human we have 46 chromosomes or 23 identical chromosomes. So let's talk about the human body cells or the 46 chromosomes. So each chromosome is composed of single tightly coiled DNA molecule. So if you're talking about chromosome and DNA, chromosomes are composed of single tightly coiled DNA, and DNA is a long threaded structure which get condensed to form chromosomes. So chromosomes cannot be seen when cells aren't dividing, and they are called as chromatin. We are going to talk about these in detail, the chromatins, when we are going to do the cell division. So here you need to understand that what is a chromosome and what is a DNA. So DNA is nothing but it's a tightly coiled DNA, which makes a chromosome, and a loose thread-like structure is DNA. So let's see how does DNA transforms into a chromosome, and that's compacting, that's condensing of DNA, which makes it into chromosome. So DNA is nothing but a tightly coiled around proteins which are called as histones. So in this structure you can see if the DNA gets condensed and it coils around some of the protein called as histone, which are the H1. So here the compacting of DNA into chromosomes. So what's the basic difference in a chromosome which are in the dividing cells and a normal cells? So when there is a duplication of chromosomes, they are called as chromatid. So here you need to understand what are chromatid. In cells which are dividing, the chromosome duplicates and they are called as chromatids. So these chromatids are held together by a centromere. So if you look at this structure, there is a center which is called as a centromere, and these are the two chromatids and also called as the sister chromatids. And as a whole, this is a whole as a chromosome which is duplicating into two sister chromatids which are attached in the center with a centromere. Karyotype. Karyotype is, you can say, what is a karyotype? It's a kind of a picture of all those chromosomes. And here is a picture of chromosomes from a human cell arranged in pairs by sizes. So you can see the first chromosome is the biggest and going to the 22nd pair, which is the smallest one. So the first 22nd pairs are called as the autosomes. So these first 22 pairs are called the autosomes, and the last one, that is the 23rd pair, is the sex chromosomes. And these are the sex chromosomes. In females you have XX, and males you have an XY. So which chromosome decides whether it's going to be a boy or the girl? So the presence of the Y chromosome decides it's going to be a boy or the girl. If it has the X and X, it's going to be a girl, but the presence of the Y chromosomes makes it a boy. So now we have looked at why the cells should divide, what are the karyotype,